Sounds good. I think we're ready.
Okay, let's do it.
Hello, everyone. My name is Hunter Howell.
My name is Reece Bawden.
And my name is Jason Blanchard.
Today, we are bringing you a presentation on the effect of intermittent cold exposure on energy consumption. So, each of us have an ideal for our bodies. We all have something we're working towards. We all have goals. For many of us, that involves weight loss or losing body fat percentage. So, in order to do that, it can be a very difficult process. The only really proven way to be able to consistently lose body fat percentage is through energy expenditure, energy in versus energy out, and that happens through caloric expenditure. So today we're going to talk about the effects of intermittent cold on that. Energy consumption, obviously, it's a huge role in weight loss and increasing our metabolic rate. It comes through our energy consumption is through our metabolic rate. So if we could potentially increase that, we could make losing weight much, much easier. The primary driver of this study is built around beige adipose tissue. That, to explain what it is versus regular adipose tissue, what we know as white adipose tissue. White adipose tissue is a type of insulation for our bodies, as well as a source of energy when our bodies are running low on drawing from other forms. It's not a super accessible form of energy, so our body doesn't like to dip into it quite as much as we would probably like it to. That white adipose tissue is has very few mitochondria. It's basically energy in the form of stored triglycerides. That versus beige adipose tissue. Beige adipose tissue has lots of mitochondria, and it's actually integral in the process of thermogenesis. So, thermogenesis is the process by which our body creates heat. And this is known as non-shivering thermogenesis when it's done through beige adipose tissue. That comes through the function of beige adipose tissue UCP1, which is also known as the uncoupling protein 1. That allows it to function at a high rate, the mitochondria to produce a lot of energy, and that energy is released in the form of heat, which consumes the adipose tissue and can be a huge driver in weight loss. The benefits of EAT is it's a very easily accessible source of energy for our body. And the awesome part is our dream body could be as close to as easily attainable as taking a cold plunge a couple times a week. As we dive into this study and the effects that has on our beige adipose tissue and our energy consumption as a whole.
Awesome. So, like Hunter said, we're going to be studying the research paper that we read is going over the effects of intermittent cold exposure on the body. So, for the researchers, for the design of this study, they came up with 3 different experiments known as intermittent cold exposure, or ICE 1, 2, and 3. Now, these are pretty similar, except they vary in a couple different ways. So, the researchers decided to study mice for this experiment and use mice and put them through different amounts of cold exposure and to measure the variables that they find from these. So in ICE 1, or intermittent cold exposure 1, we have 3 groups. Each have 8 mice that are all the same weight. The groups have the same mean weight. And we have our control group, which is not going to be put into any cold exposure. And then we have our experimental 1 group, which is going to be put into 4 degrees Celsius 3 times a week, Monday, Wednesday, Friday, for 1 hour. And then our third group of ICE 1 is going to be put into 4 degrees Celsius, that cold exposure, for 4 hours, Monday, Wednesday, Friday, so 3 times a week. Now, the second experiment, ICE number 2, we're going to have the same 8 mice in each one of these groups. We're going to have our same control group, but then our experimental group, our first one, is going to be in there for longer, for 8 hours, or for 4 hours, 3 times a week, Monday, Wednesday, Friday. And then our third experimental group is going to be in there for 8 hours, Monday, Wednesday, Friday, in that intermittent cold exposure. Now, our ICE 3 is where it gets a little bit more complicated. They wanted to introduce a synthetic compound known as AM251 and look at its effect when it is added with this intermittent cold exposure. Now, if you're like me and you heard AM251 and you have no idea what that is, that's exactly how I was, so I'm going to explain briefly what it is. So the AM251 is a synthetic compound that acts as a selective antagonist of the cannabinoid receptor type 1, or CB1 receptor. It was developed as a research tool to study the physiological and behavioral effects of endocannabinoid and the cannabinoid system in animals and humans. CB1 receptors are mainly found in the brain, where they play a key role in the modulation of various physiological processes such as appetite. So for this experiment, in simpler terms, the AM251 acts as a CB1 antagonist that both reduces food intake and increases metabolic rate. So for our 4 groups that we're going to see in our intermittent cold exposure, our third group, we're going to have 2 control groups. One is going to be given just a vehicle through which, well, the vehicle is just a substance that is used to dissolve or suspend the AM251 to the mice. So we're going to have our control group that is going to be admitted to just the vehicle, our control group that is going to be admitted to the AM251. Then we're going to have our ice groups, one of which is just going to be exposed to vehicle, and the other one just is going to be exposed to the AM251. Now, the intermittent cold exposure, the 2 groups in the ice 3 group, they are going to be in cold exposure for 4 hours, 3 times a week. So these are going to be our 3 groups, and that's how. Oh, and in ice number 3, we have 4 groups of mice, like I said, and we're going to have 6 mice, each same mean weight in each one of those groups. And if you want to switch the slide over to what we're going to be measuring. Now, in every one of these environments that these mice are kept in, they are given an unlimited amount of food, which means they can eat based on what their body is telling them that it requires. So in ice 1 and ice 2, we're going to be measuring the body weight of the mice, the amount of food intake that they eat, and their body compositions as far as fat mass and fat-free mass. In ice 3, we're going to be measuring the same thing, the body weight and food intake, but instead of measuring like in ice 1 and ice 2, they just measured the body weight and food intake on those cold exposure days. We are going to be measuring the body weight and food intake daily prior to the admission of vehicle or AM251. And we're also going to be measuring the body composition every 2 weeks, fat mass and fat-free mass of these mice. So the variables that we're going to be looking for in our results are going to be that body weight, that food intake, and that body composition as a result of the intermittent cold exposure, the amount of it, and the intermittent cold exposure added with the AM251 synthetic compound.
Okay. So for our results, essentially this graph or these graphs are showing that despite the intermittent cold exposure, there was no significant change in body mass because the intake intake was fully compensating for the loss of calories burned during the cold exposure. On the left side, we have graphs A, and on the top represents ice 1, and on the bottom represents ice 2. On our right, we have graphs B, top as well, ice 1, bottom, ice 2. Graphs on the right represent the body weight, and graphs on the left represent the food intake. We can see that over the 10 weeks period after the exposure to cold, the mice, their intake of food did increase compared to the control on all scales, every week without fail. And for the body weight, it kind of maintained and increased at the same time. So for our next graph, we wanted to focus on energy expenditure versus time. So right before the mice were exposed to cold, they were given a selective beta-3 agonist to help us estimate their energy expenditure. This helped us measure whether or not they were burning and how much calories they were burning. So we can see that for ice 1, the group that was exposed for 4 hours had the most energy or energy expenditure, and as well as the groups in ice 2. Our next graph was taken from a IPGT or GTT, which is a glucose tolerance test. This test was to see if the exposure to the cold did increase the metabolic rate. For ice 1, this test was ran the day after they were exposed to the cold. As we can see, a normal blood glucose or IGTT curve should look like the control. Anything below that curve shows an increase in a metabolic rate. The ice 2 group, their test was ran 2 days after their exposure, and we can see that all of those look pretty normal. And so basically, the results from this test show that there are temporary benefits to cold exposure on our metabolic rate. And for our last set of graphs, these were all the tests that we had ran with, but these ones were ran on the ICE 3 group, and they were given the AM 251. So for graph A and B show that AM 251 did in fact lower the intake of food and more or less suppress the appetite of the mice. As well, in graph D showed that AM 251 did in fact increase the metabolic rate. However, though, on graph C it shows that the drug AM 251, in addition to the cold exposure, did not work in a synergistic effect as they were hoping. As we can see that the mice treated with the drug had a lower energy expenditure than the control mice, saying that it did not quite have that effect on energy expenditure. They weren't eating as much and they weren't burning as much. It was just kind of a neutral effect that the mice had on the AM 251.
So what we concluded from this study is that there's a very strong correlation between intermittent cold exposure and the activation of BAT, as well as just an increase in caloric expenditure. It's very possible that this, combined with a diet, with a dietary intake, a regulated dietary intake, could very possibly make it easier to lose weight. Losing weight could be as easy as just taking that cold plunge and increasing your energy expenditure. Thank you so much.
Thank you.
Sweet.